Hello everyone, welcome back to our channel. In today's class, we are going to learn about an important process in human digestion known as digestion of proteins in the stomach. Now, whenever we eat foods such as eggs, milk, meat, fish, pulses or soya beans, they contain proteins. But have you ever wondered how these large protein molecules are broken down into simpler substances that our body can absorb and use? Well, the answer lies in the stomach and the gastric juice it produces. In this video, we will understand the functions of the stomach, the components of the gastric juice, the role of hydrochloric acid, activation of gastric enzymes, and finally, how proteins are converted into proteoses and peptones. But before we begin, a quick suggestion for students. If you are watching this video for revision purposes, you may increase the playback speed to 1.25 or 1.5x. However, if you are learning this topic for the first time, I recommend you watching at normal speed. So, let's begin. First, let us discuss the functions of the stomach. Many students think that digestion starts and ends in the stomach, but actually, the stomach performs several important tasks. The first function of stomach is to store food. After a meal, the stomach can store food for approximately 4 to 5 hours. This is temporary storage which allows digestion to occur gradually and efficiently. And the second important function is mixing of food with the gastric juice. The muscles of stomach continuously contract and relax. These movements are called churning movements. Think of it like a washing machine mixing clothes and water together. Similarly, the stomach mixes food with digestive juices through churning movements. As a result of this mixing, food becomes a soft semi-liquid substance known as chyme. So remember, food plus gastric juice plus churning movements equals to chyme. And most importantly, the stomach creates the right conditions for protein digestion to begin. Now that we understand the functions of the stomach, let us discuss the components of gastric juice. Gastric juice is a digestive fluid secreted by the stomach and it contains several important substances including mucus, bicarbonates, hydrochloric acid or HCl, pepsinogen and prorenin. Each component has a specific role. So let's understand them one by one. First, let us discuss the role of mucus. The stomach produces a strong acid capable of digesting food. But a question arises. If acid is so powerful, why doesn't it digest the stomach itself? The answer is mucus. Mucus forms a protective coating on the inner wall of the stomach and it lubricates the food and protects the stomach lining from damage caused by acid and digestive enzymes. In simple words, mucus acts like a protective shield. Next, we have bicarbonates. Bicarbonates are basic in nature. They help to neutralize excessive acidity inside the stomach. This buffering action protects stomach tissues from acid damage. Thus, bicarbonate helps to maintain a balance within the stomach environment. Now we have the most important components, hydrochloric acid or HCl. Hydrochloric acid creates a highly acidic environment with a pH of approximately 1.8. This acidic condition is extremely important for protein digestion. HCl performs several functions in which the first function is it provides the optimum pH required for the enzyme activity. Second, it destroys the harmful microorganisms that enter the body along with the food. And third, most importantly, it converts inactive enzyme precursors called proenzymes into active enzymes. This function is very crucial because digestive enzymes must first be activated before they can perform their work. Now let us see how these enzymes become active. Let us now discuss the activation of gastric enzymes. The stomach secretes certain enzymes in inactive forms. These inactive forms are called zymogens or proenzymes. Examples include pepsinogen, prorenin. These inactive forms prevent the stomach from digesting its own tissues. So, when hydrochloric acid is present, these inactive enzymes become activated. Let us see how. First, let us see pepsin formation. Pepsinogen is an inactive enzyme. When it comes into contact with hydrochloric acid, it gets converted into active pepsin. And interestingly, once some pepsin is formed, it can activate additional pepsinogen molecules. And this process is known as autocatalysis. Therefore, pepsinogen plus HCl produces pepsin, and pepsin helps to produce even more pepsin. Now let us discuss renin formation. Prorenin is another inactive enzyme found in gastric juice. When exposed to hydrochloric acid, prorenin is converted into active renin. Renin plays an important role in the digestion of milk proteins. This enzyme is especially significant in infants who consume milk as a major food source. Now let's understand how these enzymes digest the proteins. First, let us discuss about the action of pepsin. Pepsin is the major protein digesting enzyme present in gastric juice. Its primary function is to act on proteins. Proteins are large and complex molecules that cannot be absorbed directly. So, pepsin breaks these large protein molecules into smaller intermediate products called proteoses and peptones. You can think of this as cutting a long chain into smaller pieces. The proteins are not completely digested at this stage, but they are broken into smaller fragments that can be further digested later in the intestines. Now let us discuss the action of renin. Renin specifically acts on casein, which is the major protein found in milk. In the presence of calcium ions, renin converts casein into calcium paracaseinate, also known as curd. After this step, pepsin acts on calcium paracaseinate and converts it into peptones. And this process makes milk protein easier to digest. That is why renin is particularly important during infancy. Now we got the final products of protein digestion in the stomach. These products are called proteoses and peptones. First, proteoses. Proteoses are intermediate products formed during protein digestion. They are larger and more complex molecules. Since digestion is not yet complete, proteoses still require further breakdown. And peptones are simpler protein digestion products. They are smaller and less complex than proteoses. These products move to the small intestine where the digestion further continues. Therefore, peptones represent a more advanced stage of protein digestion. Before we conclude, let's understand the complete process in a simple flow. Food enters the stomach stores food and it churns it. Food mixes with gastric juice. HCl creates an acidic environment and activates enzymes. Pepsinogens become pepsin. Prorenin becomes renin. Pepsin and renin act on proteins. Proteins are converted into proteoses and peptones. Now let us quickly summarize today's lesson. The stomach stores food and mixes it with gastric juice through churning movements. Gastric juice contains mucus, bicarbonates, HCl, pepsinogen and prorenin. Mucus protects the stomach lining while bicarbonates prevents excessive acidity. HCl creates the acidic environment needed for digestion and activates digestive enzymes. Pepsin digests proteins into proteoses and peptones while renin helps to digest milk protein. Therefore, protein digestion in the stomach occurs mainly through combined action of HCl, pepsin and renin. Thank you for learning with NutriZoo Learning. If you found this lesson helpful, don't forget to like, share and subscribe. See you in the next class.